and for obvious reasons, I'm speaking on this slide. So I'll be here today talking to you about coral dispersal patterns and what insights that can give us about connectivity of corals. This work has been done with Andrew Baird, Saki Hari in Okinawa, and Sean Connolly. So today's talk, I will first say something about what we know about the current dispersal patterns of corals and what are the mechanisms that drive those dispersal patterns. And then I will elaborate more, and the main focus of my talk today will be about the future dispersal patterns of corals. This is some work that I've been doing and is currently under review. So what we know about coral dispersal patterns is mainly based on what genetic studies tell us. And this is just one citation from a paper, but there's a lot of papers showing this. And what they say is that there are considerable genetic subdivision between populations. And this happens for all species. And actually what they infer is that larval dispersal is surprisingly limited. And that is true even for species that have a long planktonic stage. So what this leads to, what this implies, is that corals have a high self-recruitment, meaning that a lot of the larvae that are released on a reef tend to settle back on the same reef, and also that they have a limited long-distance dispersal. And by limited, I do not mean that they do not disperse long distances, but just that there's a very small number of larvae that actually disperse very long distances. So the mechanisms that explain this coral dispersal patterns, of course, currents is one of the basic things. But the reason why we see that there's a high self-recruitment of the corals is because a lot of times larvae get retained behind the reef in eddies, so small-scale dynamics that retain and trap the larvae in the reef, and therefore make the larvae stay in this reef for a much longer period of time, and eventually they might even settle there. Another mechanism that allows larvae to self-recruit a lot and also determines how much they disperse is their larval competence dynamics. So broadcast spawners release larvae into the water column, which then fertilize and go through all the embryogenesis while they're still in the water column. And after a certain time, these larvae do become competent, and meaning that if they have a good substrate, they will settle and metamorphose and become a coral polyp. If they stay in the water column for a really long time without finding a good place to settle, they just lose the ability to settle. And so despite the fact that they're alive, and they can be alive in a plankton for a long time, they just are completely irrelevant for recruitment purposes. So this period here, where larvae are developing in the plankton, varies a lot between species. And even among broadcast spawners, some species can take 1 day to develop and start settling, other ones can go up to 5 days. But it's not very long, as some other models actually tend to assume that larvae take around at least 7 days to do it, and that's just not true. But brooders, in particular, this is particularly true for brooders because they do the embryogenesis inside the coral, and they release larvae that are already competent to settle. So therefore, they have very high chances of settling back on the reef where they were produced. So I guess what I've shown in a paper that I published early this year in ecology was that if we actually accurately represent this physical retention of larvae around the reefs, and if you accurately represent the larval competence dynamics, you're actually able to predict really high levels of self-recruitment, which are coherent with what the genetic studies tell us that exist. And this is really important because a lot of the existing models of coral dispersal do not show this and actually underestimate heavily high self-recruitment and overestimate long-distance dispersal. But the reason why I wanted to talk to you about these mechanisms is that one of the mechanisms here is the larval competence dynamics. And as the climate warms, we expect that the increase in temperature will affect all these developmental rates and also survival rates. So I wanted to answer to this question. So how will coral dispersal patterns, particularly, I'm going to focus on self-recruitment, how will that change as temperatures warm? And the result is not obvious. So what we know is that as temperature increases, usually larval development accelerates, and larvae should be able to settle earlier on. So while before larvae might be flushed away from the reef, in the future, they might still be able to settle before being flushed away from the reef. But on the other hand, we know that as we increased temperature, usually survival goes down. And therefore, that should reduce the overall recruitment of the species, and of course, also self-recruitment. So what we really needed to do is to quantitatively assess these two effects of temperature to figure out which one of them is stronger and in what condition, to know what's going to happen to the dispersal of corals in the future. So the approach that I used was to develop a model that has a model of larval retention around the reef, and it incorporates the competence dynamics of the larvae. And I calibrated the effect of temperature in this model. And then I used the model to project what will be the self-recruitment in the future. So the model is relatively simple. I pretty much follow the number of pre-competent larvae that are in the reef where they were born. And as you can imagine, that number goes down as larvae get flushed away from the reef, as larvae die, and also after a certain time that they need to be in the water column to complete their development, they will then, the number of pre-competent larvae will reduce because they'll acquire competence and they will settle. So the model has 4 parameters. One of the parameters is the flushing rate. On top one, I used values that were measured by Kerry Black for the Great Barrier Reef. He measured these values in reefs with different sizes and different shapes. And this range of flushing rates that he uses, and this is an exponential decay of particles in a reef. This range of flushing rates that he gives result in retention times around the reef from just a couple of hours to everything like 16 days. So I think it does give you a good idea of what might happen in different reefs. The other parameters are the biological parameters that I estimated using larval experiments in a lab. So I started by just doing this for Acropora millipora. This was collected in Orpheus. And I reared the larvae at 3 different temperatures, 27, which is their ambient, and then 29 and 31. So I followed a certain number of larvae over time. Every day I counted them and obtained the survival curves after different temperatures. And then I did settlement experiments. So I first had ceramic tiles that I put out in the ocean in a place where the adults occur. I left the tiles there for 2 months, let this ceramic tile to get covered in all sorts of coral and algae that are good settlement cues for the larvae. And then with the larvae that I had that were just born since day 1, I put on this tile inside the jar. And every day I removed the tile, counted how many larvae had metamorphosed or settled, and then put a new tile in. So with this, I was able to get the frequency distribution of settlement at the different temperatures. And of course, estimate my physiological parameters, which are the mortality rate, the per capita rate of acquisition of competence, and the minimum time to competence. And not surprisingly, I found a strong temperature dependence of the survival and competence parameters. So if you can see here, in Acropora millipora in survival, they die a lot in the beginning at any temperature, but they certainly die much more at 31 than at 27. And then in terms of competence, so how fast do they acquire competence, here what you have is the time that they stay in the plankton. And as you can see, at 27 degrees, they take around at least 4 days to start settling. At 29, they take just about 3 days. And at 31 degrees, they take a little over 2 days. So pretty much if you increase temperature 4 degrees, they almost halve the time that they stay in the plankton. So that's a really strong effect. Of course, then I use these things that I have quantitatively assessed to project what's going to happen to self-recruitment. I'm just first going to explain you what the graph is and then show you the results. So here I have the increase in self-recruitment relative to current day conditions. So the line 1 meant like that self-recruitment would not change. Of course, values on top of it meant that self-recruitment increases versus 2 meant that self-recruitment doubled. And below the line means self-recruitment decreases. And I'm doing this for reefs with different retention times. So anything like from reefs that larvae get just get to stay there for a few hours to reefs where larvae get stuck there for 20 days, and just to see how will this change depending on the size of the reef. So this is my results, and I think it's very interesting to see is that in reefs that have short retention times, you actually see a significant increase in self-recruitment. And this is because in these reefs, the larvae of Acropora millipora usually get flushed away from the reef before being able to settle. This is what happens nowadays. But in the future, they will be able to settle while they're still in the reef. And this will be a stronger effect than the increase in mortality for this species. On the other hand, on reefs that have longer retention times, you'll see a decrease in self-recruitment. And this is because in these reefs, even nowadays, larvae already can settle when they are in a reef. So an acceleration of development is really not going to change a lot how many recruits you have. What is going to have a bigger impact is the fact that larvae die more at higher temperatures. So in these reefs, you actually see decrease in self-recruitment. But I wanted to know what are these reefs where you're actually going to see a high self-recruitment. And it ends up that most reefs around the world have very short retention times. So they have retention like Hawaii, French Polynesia, Japan, and even in Australia. Retention times are usually of a few hours to up to usually 5 days. Of course, there's places that have higher retention times, areas like lagoons and stuff. But most reefs do have actually a relatively short retention time. And therefore, I guess I can say that in the large majority of reefs around the world, we will actually see an increase in self-recruitment of Acropora mellipora. But you know, Acropora mellipora is just one species, and what does it really tell you what's going to happen with all the corals? So I wanted to see if climate change affects all species equally. And so I went and repeated this experiment in Okinawa, and I used other 4 species. I still have Acropora mellipora there, and to compare the results. And I chose these species in particular because they have very different egg sizes. And this range of egg sizes kind of represents almost the full spectrum of egg sizes in corals. There's some that are a little bit smaller than that still. But, and what I know is that the egg size is a very good predictor of what happens to the size of the larval development time. So what we know is that species that have small egg size usually develop quite fast and can settle very fast. And species that have big egg size usually need to stay a long time in a plankton before they're able to settle. So I'm expecting that species that have different egg sizes will be differently impacted by temperature too. So this is my final results and the comparison of all the species. And as you can see here, I have here the species that have the smallest egg size is a fungid, and all the way to the favids and the acroporas and the lobophilia will have the bigger egg size. And I think it's a clear trend here that, remember, above the line, it means increase in self-recruitment. That the bigger the egg size, the bigger will be the increase in self-recruitment. And also that species that have a small egg size system develops so fast already. When you accelerate temperature, when you increase temperature, you only accelerate their development for a couple of hours or so. So that doesn't really affect their dispersal. What's going to have a bigger impact in those cases is the increase in mortality due to the temperature. So actually, the fungid actually even had a reduction of self-recruitment. But most species, and I have to say that the most common species in the reefs, the favids and the acroporids, do show a significant increase in self-recruitment, double in some places and even 3 times and 4 times more in other reefs. So it's a very significant increase in self-recruitment. But I didn't want it to finish here. Just the large majority of reefs around the world, you'll see an increase in self-recruitment, but this will be particularly relevant for, of course, species that have big egg sizes. And I think this is a really important information for management purposes. And I wanted to explore a little bit what this might mean in terms of management. So when you have a really high self-recruitment, one of the things you obtain is a very good relationship between the number of recruits you get in a reef with the number of adults that was on that reef. So if the adults in this reef get disturbed, you're likely to see a reduction of the recruits in this reef. At the same time, if you protect the adults on this reef, you might be able to sustain a really high level of recruitment. So it does open the door for some sort of local management actions. Hopefully, local management actions might have become a bit more effective. Trying to see here the bright side of all this, of course. And I think not so bright side of this is that as you increase self-recruitment, so more larvae stay on the reef where they're produced, means that less larvae get dispersed, right? So even if these larvae that get dispersed, if they still die more, means that less larvae actually will disperse and get to other reefs, and that will weaken connectivity. I'm not talking here at the moment still about distances that the larvae will do, just about the number of larvae that will do long distance dispersal, which is already low, might become even lower. And that, of course, might have effects in what it might have impacts for management, because, for instance, you might have to shift or reduce the spacing between marine protected areas to keep the same levels of resilience we have nowadays. And I think that's my talk. I finish up with, you'll see this is fungi larvae. So I just wanted to finish here. Thank you very much all.